Okay, we'll go ahead and start by taking a moment to acknowledge the place in which we are or would have been and will be soon again gathered. The lands and waters currently occupied by the University of California, Davis hold the stories of Indigenous peoples from time immemorial. We acknowledge the ancestral and present relationship and responsibilities to this place for Patwin and Miwok peoples, which is now home to many peoples and nations. We ask that you also take a moment to consider the Indigenous places on which you reside, wherever that may be, and offer a sense of gratitude for the Indigenous peoples who stewarded your lands and waters long before we all arrived. So thank you. And welcome to the first of four Radical Fermentation Dialogues. My name is Jessica Perea, and I will serve as our introductory MC for this webinar. I'm a Dena'ina Alaska Native musicologist and assistant professor of Native American Studies at the University of California, Davis. My own research centers Indigenous-led and indigeneity-centered approaches, methods, theories, and praxis to research, teaching, and service. I specialize in music, sound, and performance studies research with Bind for Indigenous peoples and communities of the North Pacific and circumpolar Arctic regions. I want to give a brief introduction to the series as well as the format for the webinar series, and after which we will turn to individual introductions from each of the cluster coordinators, followed by the talk and brief Q&A at the end. So the series in the cluster brings together the coordinators of a Davis Humanities Institute transcollege research cluster we're calling Radical and Relational Approaches to Fermentation and Food Sovereignty. And we're hoping to engage interdisciplinary dialogues about fermentation science, Indigenous food security and food safety, performance, feminist science studies, and radical relational approaches to fermentation. The goal of these webinars is to offer explanations of each of our individual research projects as they intersect with these topics, and to explore the relationships between and amongst our seemingly disparate research areas. We are pleased to welcome you to join us in the creation of this research community and look forward to hopefully engaging some of your questions by the end of this webinar or in person in a not too distant future. And so a little bit about our cluster, which you can find out more about if you haven't already visited our in progress website, which is Radical Fermentation at ucdavis.edu. But our cluster centers Indigenous communities and the ways they regard traditional foodways as core components of community health and wellness. And yet traditional foods such as those made by fermentation practices are threatened by settler capitalist nation states that continue to legislate away Indigenous peoples' rights to ancestral lands and waters. The goals of our transcollege research cluster includes curating a year-long series of convergence research activities, which these dialogues are the first of our public-facing events that center radical and relational Indigenous knowledges and ways of fermenting foods. Ultimately, our cluster seeks to unsettle and expand dominant modes of knowledge production in food science research in ways that advances food sovereignty, an issue of urgent global significance for all peoples. And through curated gatherings, guest expert presentations, we'll have more of those next quarter, participants in our research community will engage in collaborative projects that demonstrate how Indigenous food fermentation practices exist in relation with rather than in control of, and how with nature, and how Indigenous scientific knowledges demonstrate deeply rooted or radical relationships between human, environment, and more than human entities. Our radical and relational analyses of fermented food and food sovereignty discourse and praxis are guided by the following questions. What is the significance of radical and relational perspectives in food science research? How are Indigenous fermented foods represented across different disciplines, and how might we critically address such subconscious biases in different disciplines? Whose stories matter and who decides? And instead of proposing singular truths or facts, our cluster invites participants and collaborators to consider the existence of multiple simultaneous truths, all of which are culturally constructed, performed, and in some cases politicized and policed. I believe we also, as of today, have a social media hashtag, hashtag radical fermentation. So be on the lookout for that. We'll be building a presence that way as well. And without further ado, I'm going to turn it over to my colleagues, Stephanie, Avia, and Maria, to give their introductions and to move into today's talk. Thank you.
Thank you, Jessica. I have a trombonist playing in my house, so forgive any trombone sounds. My name is Dr. Stephanie Maroney, and I am a feminist science and food studies scholar that is interested in creating collaborative projects on both fermentation and mycology. So my current research explores fermented foods and mycelium as methods for making, repairing, and caring for knowledges and relations. I have published articles broadly on the topic of the sociocultural impact of human microbiome science, with specific articles on the queer fermentation praxis of Sandor Katz, the colonial afterlife of microbiome sampling projects, and healthism in probiotic dietary culture. I have a PhD in cultural studies from UC Davis, and I currently administer the Mellon Public Scholars Program here, which is a community engaged arts and humanities research program for graduate students. I'm really excited that you all are interested in this topic, and I hope to have more dialogue with you. And I'll pass it over to Aviaja.
Thank you, Stephanie. My name is Aviaja Hauptmann. I have a background in microbial biotechnology from the University of Copenhagen. I did my PhD at the Technical University of Denmark in microbial metagenomics. And currently I'm only virtually, but still a visiting postdoc at Maria Marco's lab at UC Davis. And for the past 3-4 years, the focus of my research has been on indigenous fermented foods of the Inuit, the indigenous population residing in the Arctic. And I was born and raised in Greenland, and I'm Inuit. So in everything that I do, also with my research, I bring that personal perspective into the research and the communication around what I do. So I'm going to keep it short. Just also say that how thankful I am to see everyone who signed up for this. It's very moving to see.
Hello, my name is Maria Marco. I'm a professor in the Department of Food Science and Technology at UC Davis. As you'll hear today, I'm going to give some perspectives on my research, which is broadly focused in microbiology and the relationships between microorganisms in fermented foods and in our digestive tracts through our gut microbiome. And with that, I'm going to start my slides. Hopefully these are all visible to you. Okay, so for today's talk, leading off with, I would say, the kind of perspective from food science and as a microbiologist, and wanted to start the conversation with the question, what is a fermented food? And so this first image sharing is of a slice of blue cheese. And I want to invite you to ask yourselves what you think of, what you feel when you see this or this image. Perhaps it's the colors, perhaps it's a sense of the taste, the aroma, an environment, the context in which we would eat this food, or perhaps a health benefit we might have, or implications in health when we eat cheese. Similarly, I want to ask the same questions here, like what do you feel, or what kind of experience do you have when you see this image of soy sauce? Perhaps it's a brand. Again, perhaps it's a taste or the context in which we consume that soy sauce with other foods. So seeing these foods and these images may evoke certain emotions or perceptions. When I see these foods, this is what I see. I see a microorganism like the one shown here, which is an electron micrograph of Lactobacillus florum that we isolated from a citrus fruit. And in this image, you see this sort of lattice work between the microbes, which we don't know what it is, but it's just one depiction of what I see when I see these foods. When I see these foods, I also see complexity. I see different microorganisms, often numbering in the hundreds of thousands, where we have different species, fungi, bacteria working together frequently, often in succession, as we see here in this with these cheese rinds being depicted in this microbial complexity image here. I also see interrelationships between microorganisms, as in yogurt, where we have two species, Streptococcus thermophilus and Lactobacillus bulgaricus, where their cooperation leads to compounds that give the sensory attributes of yogurt. Their interactions can also be inhibitory to one another. And then you can see here in this diagram how that relates to their environment, the milk environment, ultimately resulting in what we know of as yogurt. Also, what I see when I see these images, I see this elegance we have from when we start with just about any type of starting ingredient, milk, meat, fruits, vegetables, grains. What we do in preparing them at the very elemental level in our kitchens by cutting them, spicing, packing them in containers, maybe heating them. And then what comes next is storing them perhaps on our countertop, putting them in special containers where we can control the temperature passively or actively, adjust the amount of oxygen this food receives, adjust the pH, perhaps through some vinegar and water, changing water activity or the amount of water a microbe will see. And then through that, what we have is that microbial growth to lead to these foods we know as fermented foods. When I see these foods, I also see just thousands of fermented foods made around the world, and the tremendous variety that we have and recognize as fermented foods, as shown in this really elegant periodic table of fermented foods prepared by Michael Gonzalo. So this brings me to our working definition of fermented food. And this definition is not new. It's taught throughout all microbiology and food science courses. It is a new definition in the sense that it is compiling different ideas through a panel of experts that met, including myself, through the International Scientific Association of Probiotics and Prebiotics last year, where myself, other experts in making fermented foods, those in medicine, immunology, nutrition, discuss this idea of what is the definition of fermented foods, and this is what we reached as fermented foods are foods and beverages made through the desired microbial growth and enzymatic conversions of food components. So the key word in all of this, of course, is microbial. But we can deconstruct it and talk about these different components we put in, for example, desired in this word, in the definition throughout our series. So with a definition, we set a framework for doing research and talking about these foods. In our discussions, in our consensus work, we framed what is a fermented, what is included. We try to be as inclusive as possible. And this is somewhat of a distinction we have in understanding biochemistry, or the metabolism of microorganisms, where we know fermentation is a metabolic pathway and respiration is another metabolic pathway. Some fermented foods do rely on fermentation metabolic pathways, but others, for example, like when we make vinegar, rely on respiratory. So this is a different mode of energy metabolism that's also used in making fermented foods. Fermented foods also include those that are fermented during the making and then pasteurized. For example, like when we make bread, baking the bread does kill the microorganisms, but we would still call that a fermented food. Similarly with chocolate, with the roasting process, there's a fermentation process that leads up to when the cocoa beans are roasted. We also understand through this what we're excluding through a fermented food. So we would say a rotting cucumber shown here, we wouldn't call that a fermented food, that we would call that spoiled. So we can already start to see that definitions of spoiled versus fermented can be contextual. But for our purposes, this would not be a fermented food. It's not desired. Similarly, we wouldn't call a fermented food food where vinegar is added. We call that pickled, and so that is just a pickled food. No microbial growth is needed. Similarly, a food that's made and which a microbe is added at the end, that would also not be a fermented food. So this definition helps set that framework for what we include when we have conversations about fermented foods. So why fermented foods? Why have these foods persisted throughout human development and civilization to what we're consuming today? And archaeological records show that fermented foods have been made for as back in history as we can record. As shown here in these images I took in my visit to China in 2019, visiting places like Suzhou, China, where the city is built around making baijiu, what's shown here in the image on the left, and using practices that have been around for thousands of years. So initially, we think the development and sustained use of fermented foods has been through their capacity to have our foods last longer. Very simple, practical reasons. They're less likely to spoil, and they travel better. So a very practical purpose that we've had. And of course, we don't necessarily need this particular feature of fermented foods when we have refrigeration and other methods of food preservation, but this is going to be one of the main reasons that they've developed and have been sustained over such a long period of time. We do know through scientific research that why this is, why the foods are less likely to spoil, why they're better preserved, and that's through these very basic facets of chemical transformation of food and exposures of foods to water, having more acidic environment, low oxygen availability, and antimicrobial properties of these foods, we know are really important features for food preservation. Another reason why these foods have been so important in human civilizations has been their safety. A very important example is we look at potable water, where through much of human history, water wasn't necessarily safe to drink, whereas fermented beverages like beer would be. An example here is shown through the water pump. Take a picture I took in London, where John Snow, an epidemiologist, was traced back the cholera outbreak in London to this water pump, and the story goes, those who were drinking at the brewery close by were less likely to develop cholera. That's just one example. And what we know today is how these foods are transformed through microbial activity to make them safer. For example, reducing the pH, or in other words, increasing the acidity of the foods, prevents very dangerous pathogens like Clostridium botulinum from growing, preventing botulism. Microbial growth can also detoxify the starting ingredients, such as in the removal of phytic acid, an anti-nutrient from flour and baking bread. Of course, another reason why these foods have persisted for such a long time in our diets is through their taste. If they didn't taste good, I don't think we'd still be eating them and practicing making fermented foods these days. So we now know also how microbes are able to change the macromolecules or fundamental components of our foods to taste something different, to change the taste, to change the sensory attributes. And depending on the starting ingredients, whether enriched in proteins or fats or starch and sugars, we're going to select for growth of different microorganisms through the process of fermentation. And based on which microbes grow, we're going to get different metabolites, different compounds that have different flavor, sensory properties, aromas, even carbon dioxide, for example, that effervescence, carbonation we have in some fermented drinks. Another example there, of course, is ethanol, alcohol, being a primary endpoint, a primary compound made. So these are some well understood, appreciated facets of fermented foods as they relate to the development of different cultures and diets around the world. But there are still a lot of questions in the making of fermented foods. And I'm sharing two overarching ideas that I'm particularly interested in, have been exploring in my research. And that is the microbial processes needed to make fermented foods, understanding how they are ecosystems, and by using ecology frameworks, we can better understand the making of fermented foods. This is particularly needed in our understanding of traditional fermented foods. However, it's also needed in the scale-up commercial production of fermented foods, which are very vulnerable to quality defects as we scale up from home to large volume production. The other overarching question that we're facing with fermented foods is their impact on health. We don't have many studies examining specific and general health benefits of fermented food consumption. And we, as I'll explain, we have opportunities to understand this at a better level through mechanistic or more detailed analyses of the microorganisms and the metabolites that they make and how they impact health. So I want to take the most of the remainder of the time here just to highlight more specifically what I mean by these two major themes. For the past 150 years, we have had a better view of what microbial life through the time of Pasteur, when microorganisms were understood to not only cause disease, but also to be beneficial through making of fermented foods. And so much of the past 150 years has been cataloging which microorganisms are present in our environment and here in fermented foods. So you can see names of genera of different bacteria, fungi, including yeast and molds, that are needed for making fermented foods. So we have a pretty good handle on which microbes are important. And we also have a pretty good handle on the numbers of microorganisms in fermented foods using methods that were developed in the late 1800s, again, around the time of Pasteur. And so what I'm showing here are some results we've obtained through our work with companies in Northern California that make Sicilian-style fermented olives from using Saviano olives, big green olives. And working with several companies, we measured the number of bacteria over time when olives were harvested early, mid, late in the season, and using, again, standard microbiological methods where we culture the organisms in the laboratory. And what we see very consistently in making of these olives is this rapid bloom, a growth of lactic acid bacteria from being undetectable to over 1 million to 10 million bacteria per gram olive within the first 30 days after those olives are put in brine and submerged. And then we see a very stable population over time. We did the same thing, but on the same olives, looking for yeast instead of bacteria. Doing this showed that yeast are also important and contribute to olive fermentation. They also grow rapidly, but they reach a much lower cell number of about 1,000 cells per gram. Very consistent. Evidence of yeast growth is also, you see in that pellicle at the top of those olives. So we could stop there and say, this is what a fermentation, olive fermentation looks like. But now we have much more advanced methods in the past around 20 years where we're able to look at the genetic contents of microorganisms to understand them better. And so what I'm showing here is those same olives, but instead of through culturing, looking at their genetic contents, where we can identify them down to what species are there. And this provides a much richer view of what's actually happening on those olives as they are being fermented. So we're able to see that even though the population number, the total number of bacteria and yeast doesn't change over time, there's still a lot happening on those olives and in the brine. Whereas in the brine, we'd see more leuconostocs, a type of lactic acid bacteria. Whereas in the olives, we see initially lactobacillus, and then towards the end of the fermentation, we see the growth of pediococcus, all lactic acid bacteria. So they would all kind of look the same on our Petri dishes, but using genetic methods, we really see there's a lot more going on. Similarly with yeast, you see a succession from Candida boydinii to Pichia over time. We would miss this. We've missed it for about 100 years, and now we're able to see it through advanced microbiology methods. We even can go deeper than that. So using a microbiota approach is what I showed you in the last slide. Using more genome or metagenomic approaches, we also know that there's such diversity, even at a species level, between these fermented food bacteria. Lactobacillus, for example, are only about 70% genetically related by DNA homology studies. And when we group them together like that, I think we do ourselves a disservice because we miss a lot of the resolution at the functional level as well. When we do that, we're essentially saying we are the same as lemurs. So we're actually more closely related to lemurs than lactobacillus strains are to each other. And just to illustrate what this means, we've been studying Lactobacillus plantarum, formerly known as Lactobacillus plantarum, in our laboratory. And we have collections of isolates of this organism from different plants, fermented leaves, fruits, so a wide variety. And what we find is that even when we isolate Lactobacillus plantarum from the same food, genetically, phenotypically, they can be quite different. So looking at them capacity to grow on different sugars, their capacity to grow under different environmental stresses, such as high levels of salt or NaCl or ethanol, really differ depending on that specific organism, which we call a strain. So there's so much to be understood in this diversity and how that plays out over the course of a fermentation. As shown here, back to my olive example, we took a strain of plantarum and we inoculated it into, added it to olives at the start of fermentation. And we measured the population of this one particular organism over time during the course of the fermentation. And what you should see here is how variable, how active, or how dynamic that one organism is in its numbers over time. Independent of other organisms, we added also Saccharomyces cerevisiae to some containers as well. And so you can just see that there's a lot going on. By the end, we weren't able to detect it any longer. So this is really new. We don't understand how these organisms interrelate with each other and the impacts on that on the final product that we would eat. Another example here is we took a single strain of yeast that a Saccharomyces cerevisiae that we know can metabolize pectin. It can break down the structural components of the olive. And we put that organism into our olive fermentations. And it changed the olives from looking what you see at the top of this beautiful green and firm olive to a really mushy olive that we wouldn't want to eat. What was interesting in that process, adding that one organism to the ferment, was that it actually wasn't a very abundant organism. Although we see higher numbers of Saccharomyces in the ferment, it was only less than 0.1% of the total population of yeast in those olives. So small numbers of very potent spoilage microbes can make a huge difference. So that's the first question, overarching vision I have, and many in the field have, how we need to better understand fermented foods. But secondly, I'm also particularly interested in how these foods can impact our health. There are a variety of targets or ideas how fermented foods can influence our health and well-being. And so I'm sharing this with you here, ranging really from digestive health to metabolic, even see interest to understand how fermented foods can prevent cancer, cardiovascular disease, improve bone health, improve our immune function, and alter mood and behavior. Well, these are really interesting and I think emerging ideas for fermented foods. We have very few human studies or crossover design, randomized controls, prospective studies even, to really make such claims. They're really limited frequently to one or two per food type. And so I give some of these examples here. I won't go over them in detail, but you get a sense for that we really have a lot of questions still on the role of fermented foods as specific foods or foods as a whole. For example, fermented fruits and vegetables, how they may contribute to any one of these factors here on our physiology and health. Perhaps exception here would be, or the one that we have the most understanding would be fermented dairy, in particular yogurt, where there's been many prospective and RCT type of studies in people, just showing here one which is not targeted to fermented dairy, but a prospective longitudinal study where people's diet and weight gain was tracked over time. And so it is notable that, where we would expect maybe fruits and whole grains can be associated with weight maintenance or actually weight loss, yogurt was also amongst those that was most strongly associated with a lack of weight gain, actually weight loss over a period of 4 years. Besides understanding the impact in populations and individual health, where we also are looking in our understanding of fermented foods is how they could do this. So how, through a process that we could do in our kitchen where we cut food, we put it in a container, we wait, and we get something in the end, something that also may taste good, how that can influence our health through what molecules, which microorganisms. So I like to think of this in three main ways that this is possible. One is through the transformation of the starting ingredients. And there's a variety of ways that this can occur. The example shown here is in a dairy fermentation where lactose, the primary sugar in milk, is being consumed and therefore removed from the milk during the fermentation process. And through that process, we could see that there's improved tolerance to dairy foods among lactose intolerant individuals. So this is just one, as I said, example of how fermentation can result in the breakdown or removal of unwanted compounds. On the other hand, second way that exactly how fermented foods can benefit our health is through the production of new compounds through microbial processes. An example here is an increase in vitamin K or quinones that are made through microbial growth and through microorganisms. And vitamin K, we know, is important for in bone density and needed for blood clotting. And again, this is just one example of a compound that can be made by microorganisms and in some fermented foods that are not present in the starting ingredients can be benefiting our health. The third way and last way I see this forming is the mechanisms or specific ways fermented foods can benefit our health can be through the provision of living microorganisms. In some fermented foods, we don't have a step where we cook them or bake them or remove the microbes. And by having fermented foods like this in our diet, we increase the number of microbes we're consuming each day by over 1,000-fold. Or in those who eat a lot maybe get even more than 1 billion cells per day. This is 1 billion cells per gram, so you can multiply by the number of grams eaten. How is this important? Well, it's possible that microorganisms reach our digestive tract and interact with our intestinal epithelium and our immune system there. Through studies that we do in my laboratory, we look at individual strains of lactic acid bacteria for their capacity to modulate how and improve a barrier function and reduce inflammatory responses, in which we see in reflective of what we see in some human studies as well. So there's this opportunity with consuming live microbes. And this also opens up doors in how we talk and think about our intestinal microbiome or our human microbiome. The knowing that we're really only 1% human if we add up all the genomes of the microorganisms that are on us at any one time. So the microbiome we live with, but we also get exposed to microorganisms in food. And so how these microorganisms interact with our microbiome could be another way in which our fermented foods can alter or influence us or our health in some way. So to wrap up, I wanted to leave with just a few parting thoughts. Firstly, particularly now during our pandemic, I think it's interesting that we have a lot of trust in microorganisms when we're making and eating fermented foods. For those who make your fermented foods at home, there is an element of trust when we leave the olives or the sauerkraut out on the counter, that it's going to be safe and tastes good. Second thought, only a few fermented foods have been extensively studied. For example, yogurt and beer. But even for those foods, there were so many unanswered questions from the strain level to the specific compounds in the fermented foods, environmental factors, varietal factors that could affect the outcome of those foods. For foods made traditionally, we have even less knowledge about even who's there, which microorganisms are there. So there's a lot left to be explored. Thirdly, despite the prevalence of fermented foods in human diets, their importance for health and well-being is incomplete. Our understanding is incomplete. Fermented foods like bread, beer, wine, coffee, chocolate, they're all staples on our shelves. So one thing that we're continually striving for in my work and in my lab is to understand how the organisms that make the food and the final food that we eat can impact us through the specific food or globally through consumption of many of these foods in our diet. Before I end, just to say that as part of another activity I'm leading at UC Davis together with Erin DiCaprio, we are collecting home ferments in an Eat Lac project. So if you're interested in that, these are fermented fruits and vegetables. Please contact me because we are wanting to understand which microbes are in our home ferments to provide that, but a richer understanding of those particular foods. So with that, I'll close and thank the members of my laboratory and the funders who've supported my work over the years. And I'll stop sharing.
Thank you so much, Maria, for this talk. I think it made the perfect introduction to our dialogue series. I'm really excited to be able to have this conversation with you around the work that you've been doing for the past years. So I want to start off where you also started off, actually, with the definition of fermented foods. So I was curious if you could think of ways that that definition might have been different had it not been scientific and industrial experts gathered to make the definition. You have this outreach project, a community science project, and you are in touch with people who make lactoferments, for instance, around the country. So if those were the people who had to make the definition, do you think it might be different in any way?
Thanks, that's a great question. Yeah, I think that, you know, when we make a fermented food, we can't see the microorganisms, right? It's a very sensory or a visual type of experience. So it is hard for me to step back from my scientific hat to think about how it would be defined, and I think this is something that we will be coming back to. But you know, I would see it as sort of preparing food and pausing and waiting until it's ready to eat. So a food that's made and left for a while. So that's sort of a common that's just a common facet of fermented foods, is we start with an ingredient or raw materials that we would then just wait and have the patience before eating.
Time, yeah, and patience. I like that. That would be interesting to have that as part of the definition. So in connection to that question, the definitions that exist about prebiotics, probiotics, and now also symbiotics that combine those two, they all have the formal requirement that there's an established link to health. And I think for the Inuit, and I believe also for many other indigenous peoples and people in general who work and make their own ferments according to some tradition, I think that that there's a big aspect of emotional and social and mental and even spiritual health connected to making foods that come from collecting or hunting and then processing through that waiting time. So that is a health aspect that's not recognized in the current definitions. Could you see that? I know you can't answer on behalf of the whole consortium, but for you and the work that you've been doing and the dialogues that you've been having around these definitions, do you see a space for those kinds of health concepts to be incorporated? And if not, do you see obstacles against that?
Yeah, I absolutely see a way forward with that. We talk about probiotics, prebiotics, symbiotics, and that we really do need to have, human studies or some basis for that. They're sort of outside, but within the context of fermented foods. So wouldn't it be appropriate to say a fermented food we have we make ourselves is probiotic, because we haven't studied that for ourselves. But on one hand, it's useful to have these definitions and to be kind of rigid maybe, and when we share them, because they can be abused. But there's certainly, we're certainly open. There's certainly room for different contexts or different relations we have with foods, and through their making, through their tradition, through their needs, in regular diets where they would be healthy. And it doesn't have to fall into the context of we define it as a probiotic, or we define it as a specific compound, but broader than that. So I think there's a lot of room, a lot of space to look at these foods in different ways.
So I noticed that in the paper with the definition from the ISAP, that there's a comment there that says that fermentation is deliberate and controlled. And for myself, working with Inuit ferments, quite a large fraction of those ferments are actually made by nature. They aren't deliberate and controlled. That examples could be the content of the caribou stomach, the most popular Inuit vegetable, and the entrails of birds and other herbivores that you eat, or even fish that roll up on the shore and lie there on the shoreline to ferment, and then you eat those. So there's no control. Do these foods still fall under the ISAP definition of fermented foods?
Yeah, they do, and I'm glad you bring this up. That word, desired, deliberate, controlled, that was the hardest part of that definition, because sort of for the reasons that you bring up, like a lot of fermented foods, whether it be Inuit or making sauerkraut on our counter, it's sort of, even though it is by nature, it is a natural process, there is some human activity that is intentional there, right? And it's the intention of burying it, or the process, the tradition of putting it in a particular place that could be really important. So it isn't an accident, it is intentional. Is it controlled? Sort of, I would say yes, not in the industrial sense, but it is controlled in the way, again, through the practice of preparing.
So still, even if it's a fish that you just pick up from the shoreline and eat, and it is already fermented, would you still say that it is there's an action required for that?
If it's not prepared, so are you saying that it would just be walking along and you find a fish and then you would call that fermented?
Yes, and the same with the stomach content of the caribou. You just eat it straight out of the animal, really. So a human being did not do anything for that. The caribou did that.
Right. So in the word desired and having microbial growth, then I would say, yeah, it could be discussed in that context. Yeah, whether it be the fish example or for the caribou, where there is the actions of the microorganisms in the digestive tract, which would also be a form of fermentation.
Yeah, I noticed that it's desired because it is still the flavor that's wanted, and so, but it isn't controlled. But I also noticed that the controlled part of is not actually in the definition. It's the desired part, and those two, I see the separation with those. Yeah. So thanks. I think I'm going to go with just another question, then maybe we can go to the Q&A for the final part. So I noticed you had this paper discussing if there should be a recommended daily intake of live microbes, which I was so inspiring to me, and it really made me see a lot of future things that I want to do in the future. And I think a lot of researchers feel that way about this research. It said that fermentation started with the transition to the Neolithic revolution, transition to agriculture, and The same is also repeated in the consensus statement, the definition of fermented foods, that fermented foods might even have facilitated the transition into agriculture. But so having in your talk also acknowledged the importance for health from fermented foods, is it logical then to assume that for most of human evolution we did not consume fermented foods, that fermented foods is connected intrinsically to agriculture? And you sort of also would know where I'm going with this, because Inuit are hunter-gatherers and we have fermented foods. So I was wondering if you have a comment about how you discuss this in the consortium, whether or not fermented foods can really be said to be connected to agriculture.
Yeah, I think there's a bit of nuance on that. That requires maybe a little more explanation. Certainly the facets of food preservation and safety could help facilitate the transition from hunter-gatherer to more sessile, where food is going to be stored for a longer period of time. So that can be through the lens we see that particular dimension, you know, that facet of this. Of course, we don't have great archaeological records going back much further in time. But, you know, frankly, as far as our diets have always had some component of fermented foods. And I think what's changed is our capacity to preserve through other ways, through fire, through heat, or preserve through refrigeration, other methods. So one thing, going back to the live microbe idea, is, what's changed over human diets is our perhaps a reduction in the number of microorganisms that we're exposed to on a daily basis, those environmentally and those that we eat. So I think that's something that we're really interested to understand. And an American diet has we consume maybe 1 million microbes per day, which is really, really poor in microbial content, whereas other diets have much larger, particularly if you're eating a lot more raw foods. So I don't think we necessarily would say there wasn't fermented foods, and I don't think that's the intention in hunter-gatherer society. It's just to make sure that nuance is clear.
Yeah. Right. So I think we have one question from the attendees, or even two. So let's see how if we can manage to go through those two. The first question is, what do you think will be the major advances in fermentation science in the coming years? What would you like to see as the most pressing areas of research for industry, for human health, or home fermenting, etc.?
I see, one opportunity, or where I see these foods going, is there's a lot of interest, at least now in the US, in fermented foods, fruits and vegetables, kombucha, to sustain that. There's sort of the practical side and sort of the visionary side. The practical side is how to make those foods consistently, and if we're interested in health, how we're able to ensure that they're healthy. For other aspects of that, it's really in how to use words like control and measured and sustained. How do we continue to make these foods in different cultures, maybe in Inuit culture, where we have a lot of climate change and issues of risks of being able to make these foods safely. So I think what we have to look at now is how we can use the modern methods in science, microbiology, and other to help preserve, sustain, and continue making of these foods well into the future.
I agree. So we have just 2 more questions, but as we're running out of time, I think what will encourage is that people follow the next 3 seminars. The one question that I think we'll be diving deeper into in the next seminars is a question stating that fermented foods have been made safely for millennia. What happens when traditional knowledge on proper making of fermented food is not passed on from generation to generation? Are they still safe? Examples in meat and fish fermentation, what's the solution? So that's definitely something that we'll be talking about in the coming sessions, and at least I will be getting into that in my dialogue in March. And the final question is, do you see a future for fermented foods to be recommended in dietary guidelines around the world? And I could just refer to your recent publication where you discuss that elaborately. So I'm not sure. So we're going to be making a newsletter, or like there's a, let me just, a list of emails for everyone who signed up. And I think we can send out links to some of Maria's recent work that actually answers this question really well. What do you say to that, Maria? Or would you like to add a comment?
Yeah, so fermented foods are represented in some national dietary guidelines. I think particularly of India, but there is definitely opportunity. And for example, in our own U.S. Food surveys, the word fermented is never used. The word pickled is used, and it makes it really difficult then to decide whether it's fermented or chemically preserved. Yeah, so I think we have a long way to go.
Yeah, but I can really refer to the work that you've published recently that to look for further discussions on this. So I think we are just about out of time. Do you have any final comment you want to make before I make some closing remarks?
No.
No. Thank you, Maria, for taking the dialogue with me and for your presentation. And thanks to everyone who participated. We're so happy that there was so much interest shown in this. And I think we didn't get to answer that really interesting question, but I really urge you to follow up on the next dialogues that will be coming in the coming weeks. And everyone who RSVP'd will be added to a mailing list, and we'll send out some links to Maria's recent work. And then the session was recorded, and we'll also be sending out a link to the recording after it's been edited. And then, yeah, again, I encourage you to participate in the next 3 sessions, where each of the remaining 3 of us will be giving a talk, and one of the others will be responding to that talk. So thanks again for participating.
Thank you.